When you look around, you will observe a dizzying variety of life, ranging from 90-foot whales to microscopic organisms darting in a drop of water to jumping spiders hunting in the grass. As over 1 million species of animals have been described till now, organizing this menagerie looks like an impossible, chaotic puzzle. It is tempting to group them by where they live or what they eat, but these superficial categories fail because they place completely unrelated organisms together based on pure coincidence. To find the true systematic position of an animal, scientists look at the architectural blueprints. They study how bodies are built from the ground up, starting with the arrangement of individual cells. In this way, biological classification acts as a chronological map, documenting the appearance of structural innovations over millions of years. The base model for this map is phylum Porifera, the sponges. While they are multicellular, they lack actual tissues, operating instead as loose cell aggregates. Because of this loose arrangement, sponges are almost entirely asymmetrical. Any plane that passes through the center fails to divide them into equal halves. A major evolutionary shift occurred when cells performing the same function began to arrange themselves into tissues. These layers introduced a new geometric layout, radial symmetry. In organisms like jellyfish, any plane passing through the central axis divides the animal into two identical halves. Radial symmetry allows an animal to interact with its environment from all sides, but it is poor for directional movement. To travel purposefully, life required a different shape. That requirement resulted in bilateral symmetry, where the body is divided into identical left and right halves in only one plane. This shape, seen in platyhelminthes, enables directional forward movement and the clustering of sensory organs at a distinct head. Flatworms are flat because they are acoelomates, lacking a true internal body cavity. Their tissue is solid from outer skin to inner gut. The next innovation was the coelom, a fluid-filled body cavity acting as a buffer between the outer wall and digestive tract. This cavity provided the internal space necessary for organs to fold and specialize, leading to the massive success of groups like annelids, mollusks, and the heavily armored arthropods. The coelom functioned as an internal sandbox, housing the complex life support systems needed to eventually move from the water to the land. As bodies grew, a structural support emerged on the dorsal side during embryonic development, the notochord. This rod defines the chordates, providing a flexible internal chassis instead of a heavy outer exoskeleton. In vertebrates, this rod is replaced by a segmented, bony vertebral column. This backbone anchored the powerful jaws of fish, supported the limbs of amphibians, and enabled the complex adaptations of birds and mammals. The vertebrate skeleton does more than support weight. It provides the secure housing for the complex central nervous systems that characterize human beings. The architectural choices we've tracked form the basis of the classification scientists use to map the animal kingdom. By tracing these branches, you can follow the transition from cellular organization through the geometry of radial and bilateral symmetry, down to the nature of the coelom. Every animal finds its systematic position based on these structural decisions, ending at its specific phylum. The resulting taxonomic map reveals the shared structural history of life on Earth, where every creature, from a sponge to a human, is defined by these specific architectural milestones.